All right, everybody. Thank you for coming today. Can you all hear me? Thank you for coming today. So today is my pleasure to introduce Jonathan Michaels. Jonathan Michaels is a postdoc in my lab. Jonathan did his PhD at the German Primate Center, and then his first postdoc at Stanford, with Hans Scherberger, and then his first postdoc at Stanford with Krishna Shenoy, and he just recently moved here to start working with us. And today he's going to show us some of his latest and greatest work, combining deep learning, machine learning approaches with large-scale primate electrophysiology, direct reaching and grasping. So enjoy the talk. Thank you, Jonathan.
Well, thanks, Andrew, for the introduction, and thanks to the department for this opportunity to present today. So I'd like to start with a short video. If the presentation will work. A bit more dramatic with sound, but so that was solving a Rubik's Cube with one hand in about 10 seconds. I don't know if any of you want to see if you can beat the record. This one's almost done. This was almost done, so maybe throughout the talk you could figure it out. But for me, this is a really interesting example of really just how amazing our hands are, what we can do with our hands, and what we can train ourselves to do. Even if 10 seconds solving with one hand is something that most of us wouldn't be able to do. But definitely very impressive, and really makes me wonder how humans are doing this, and wondering how we can understand better how humans control our hands so well. So if we take another example, here's a machine developed by OpenAI to do pretty much the same thing. Here you can see it's flipping the cube around in its hand and trying to rotate the faces. So this robot can solve a Rubik's Cube in one hand in about 4 minutes. So definitely a lot slower than the 10 seconds, but probably a lot faster than some of us. And if you watch it, you can see that the behavior doesn't look quite what you would expect from a human, but if you watch, you can see it actually is getting somewhere and rotating the faces. And this represents something close to the state of the art in the field right now for this kind of task. There you see it flip the face. And eventually it does, if we wait long enough, it actually does get there. But the key here is that grasping and manipulating things with our hands is really a really difficult problem. And even though lots of people have tried to solve this and seen actually tons of success, and this in many ways is quite impressive, we haven't reached human level control. So maybe we need to understand a bit more about how humans do this. So how does this actually work in this example? Well, I'm going to give you a one-slide primer on how deep neural networks work. This was powered by a deep neural network. So here's the example for this exact algorithm. So on the one side, on the left side, what we see here is a convolutional neural network, which some of you may have heard of. So this is a neural network that takes images, in this case 3 images, different perspectives of the cube, and is filtering these through convolution. So what that means is there's some filter, so some set of weights that is multiplying by pixels in the image, and it's moving this filter along the image, multiplying by the pixels, and trying to detect specific features in the image. And you can do this for many different feature maps, so basically different sets of weights that learn different filters on the images to extract features like edges, as I mentioned. And then you can go through successive layers, also decreasing the dimensionality of that, increasing the receptive field, to try and figure out what's going on in the image. In this example, the network is trying to figure out what pose the cube is in and what the orientation of the different faces are. On the other side, we have here a recurrent neural network. So for a convolutional neural network, if you present an image, you get an output. Every time you present the image, you get the same output. There's no time involved, there's no memory involved. A recurrent neural network basically has a memory component. So the internal state of the system is evolving over time, so it has a memory and is able to produce changing inputs over time, even giving the same input. So And that network is responsible for producing the required movements of the fingers to do the actions we want to solve the cube, and it's also getting information about the fingertip position. You saw little sensors on the fingertips. Those were to tell the network where the fingers are. So actually, if we look at these components, they look a bit familiar also from a biological point of view. The convolutional neural network seems a bit like what you would want a visual system to do, maybe, and the motor system seems like what you would want, an output system that's controlling actions to do, and these fingertip locations seems a bit like, proprioception, body sense, where is our limb right now? And actually, the convolutional neural networks especially are directly inspired from a mammalian visual system. Without the study of how our visual system works, these networks wouldn't have been created. So just to give a couple ingredients for what this looks like on a general scale, I see it as kind of 3 knobs that we want to control to figure out what our neural network should do. So for example, a task to optimize. So for this network, the cube pose and the face angles. And for the recurrent neural network, the actions that are required. We need to have a particular architecture of the neural network. So in this case, a CNN combined with an RNN. And we need some training algorithm. So the most common training algorithm is backpropagation, which is basically an algorithm for finding out how we can, in small steps, make changes to the weights of such networks to optimize the specific task that we want. And all the examples I'm going to show you after this one, use backpropagation. This one also used something called reinforcement learning, which is something you want to use when you know the goal, but you don't know exactly what action you want next. So you need some way of figuring out the optimal action, while backpropagation is basically a way of getting it to output exactly what you want. So what's a deep neural network? Well, a deep neural network is really simple definition. A network is deep if it contains at least 2 layers between the input and the output. So between the images and this final output here, if it contains at least 2 layers, it's considered deep. And normally they contain much more than 2 layers. And for the recurrent neural network, it actually contains potentially infinite layers because as we progress through time, each time step is an additional layer. So this was my kind of 1-slide intro to deep learning for our purposes. And maybe you weren't completely impressed by that robotic performance. It's not at human level, but there really have been impressive examples of deep learning applied to problems in the real world, reaching human level or beyond. So one example here, using CNNs, which is really one of the most popular right now, is an example of image segmentation and classification. So CNNs are really good at image classification. This network in particular is finding objects in the scene, for example, cars or people or traffic lights, and outputting what it thinks they are. And obviously this could be very useful for something like a self-driving car, or really anything where you want to analyze images or video. And these have been very successful and reached human levels in many ways. Another great example is speech to text. So if you take out your phone and say, hey Siri, and dictate something, it actually works very well right now. And the more modern approaches to this have been based on deep learning, and people have been working on that for quite some time, and it works very well now. And perhaps one of my favorite examples that is also very well known is AlphaGo or AlphaZero, which is a program which was developed to beat human players at Go, which was something that was considered almost impossible or not possible for at least another 10 years. This is the example of AlphaGo playing Lee Sedol, who was one of the best Go players in the world, and beat him. And what I find really interesting about this example is that people actually study how AlphaGo and AlphaZero play Go now. So actually it seems like because of the solving of this problem, people are learning something new about the game that's been around for, I think, thousands of years, potentially. And what's interesting about that is that maybe if we can learn things about games like Go, maybe we can also use these tools to learn something about neuroscience. So how I think that works in practice, to give you a concrete layout of how we might do this. So this is something like how we would normally do science, right? Very basic. We have a research question, we develop a hypothesis. We do some experiment, we have some controlled variables, things we want to keep constant, some knobs for our independent variables, things we want to control carefully to test. We might get some behavior out of that, and maybe we'll have some sense of what the internal state of our experimental subject was. For example, maybe we record muscle activity from a human during the task. We might compare to our hypothesis, draw conclusions, revise hypothesis, and go on. So how I see this working in general for modeling, which doesn't just apply to neuroscience at all, is really the huge benefits of modeling are, in addition to our experiment, we also have a model which reflects the best idea we have about how the system works. And the benefits here are these four. So we have precise manipulation of controlled variables in a model. In the experiment, we try to control for everything, but maybe there are some variables that we aren't able to control in the lab setting. We have greater control of the independent variables. So there are knobs we can turn that we might actually not even be able to do feasibly in an experiment. We can collect many more observations. So instead of running a new participant, you can maybe run some code and try something so you can iterate much more quickly and collect more data. And of course, we have full access to the internal state. So here we could only measure some measure of what's going on inside the person. For the model, we have everything. Any level of it we want to analyze, we have full access to it. So this is a really general point about modeling. So specifically, how does deep learning come into this? I think there's two specific benefits. So one is that we have the ability to test complex and realistic neural structures that parallel more what we think is going on in the brain, and the ability to train these more complex structures, and going with that, the ability to test more realistic and complex behaviors, such as grasping. So if there's anything that I want you to take away from the talk, it's that I think there's a very clear benefit of using deep learning in neuroscience that goes beyond modeling, and I'm going to try to convince you of that as well. So just to give some quick examples of how this has already been used, how deep learning has already informed neuroscience. So if we take our knobs, we take a task to optimize, for example, object classification that we already mentioned, and we take a neural architecture, so the convolutional neural network. So what that looks like in practice, I mentioned that CNNs are heavily inspired by the mammalian visual processing system, specifically the ventral stream, where many people think one of the goals is to identify what's in an image, for example, the elephant here. And that's what the ventral stream looks like in primates. So information going up from the retina all the way to the IT cortex, with some feedback going down. And most CNNs parallel this design with a sequence of convolutions and then reporting an object and trained to report that object. So some short examples of where this has been useful in neuroscience are, and this has been shown in many different ways, but it's been shown that progressive layers of CNNs modeled for object classification match neural activity along the ventral stream in primates, in humor and fMRI, and many other experiments. So that means that if we look at what the activity is in this layer and we compare it to layers along the primate ventral stream, we actually see there seems to be a correspondence going up this hierarchy, which tells us that maybe something about this architecture and optimizing this specific task actually tells us something about how our brain and primate beings might be doing this task. Another example is that if you include these feedback, these top-down signals that we know are in the circuit into CNNs, you actually see that the CNNs get better at explaining human responses to difficult images, where maybe there's occlusion in the image or there's something ambiguous about the image. And one of my favorites is that if you build models like this and try and predict activity here specifically in V4 from the model, and then ask the question, for a given neuron in V4 that we're recording for, can we build a new image from pixels that will make that neuron fire more than it normally fires and other neurons less? So can we kind of manipulate individual neurons directly through vision? And they're able to use these networks in this science paper last year to go all the way through their network back to the pixels and optimize images that make certain neurons fire more or less. So here's an example for one specific neural site. And that's really interesting because it gives you an idea if your model is doing what you think it's supposed to do, it models the areas you think, and it gives you an idea of what kind of stimuli these areas care about. And that's also an approach that could be extended to other things. So in parallel, to give quick examples for recurrent neural networks, if we take the task of controlling muscles using recurrent neural network, that might look something like this. So if we think about motor cortex and premotor cortex, what they're doing in the primate, you might make a recurrent neural network that for a desired movement, like a specific movement direction, has to generate the muscle commands, for example, for the arm necessary to reach in that direction. So two examples where this has been really useful is that looking at how recurrent neural networks come up with these signals that we know the arm uses has shown that the RNNs actually support certain theories that people have come up with motor cortex and not others. So it allows us to actually test the plausibility of particular theories above others. Another interesting study showed that if you change the biomechanics in a simulated arm, only certain configurations of those biomechanics were actually able to explain what's going on in motor cortex. So actually by changing the architecture of the arm, they were able to see which ones are most consistent with what we think is going on in motor cortex. So if we take what we know from these types of architectures and these types of experiments, how can we apply these to grasping? Well, grasping is a task that we need to go from vision of an object, extract features about that object, and then come up with the right muscle commands to grasp them. So I actually think that it's a perfect opportunity to combine these types of networks and these types of questions across multiple areas, specifically in the primate where most of this work has been done. So to address that, looking at the primate, this is what the major players in the primate grasping circuit look like. So we mentioned primary motor cortex already, which has the most direct projections to the spinal cord controlling the muscles and shows strong modulation during movement. There are two other areas which are very important for this. So AIP in parietal cortex receives information from the ventral stream and the dorsal stream and is very selective for the shape and orientation of objects that are going to be grasped. As well, area F5 in premotor cortex is very selective for the type of grip required to grasp object, but also these features as well. So there's actually great similarity between these two areas. And AIP and F5 are very strongly connected, and M1 and F5 are very strongly connected. So one thing we know is that lesions to any of these regions lead to severe deficits in shaping the hand for grasping. So lesions to M1 can completely abolish movement of the fingers completely in many cases. Lesions of AIP and F5 tend to show different results. For example, removing the ability to shape the hand properly. So maybe the hand reaches out in a way that's not appropriate for the object, but in many cases, once you make contact with the object, actually then you're able to manipulate it, but you can't shape your hand properly. So one question is also to understand how these areas differentially contribute to that eventual movement. So to address this, we designed a many objects grasp and lift task for macaque monkeys. So in this task, monkeys sit in front of this turntable, which is a motorized turntable. So every trial it turns around and they get a new object in front of them. This is a set of many objects, each on a different turntable, which you have to switch out every once in a while. And on a trial, basically, they sit in the dark, and then for a short time, they get to see the object they're going to grasp. Then it goes dark again. There's a memory period, and then after a go cue, they have to reach in the dark and lift this object. At the same time, the monkey's wearing a little glove, which is recording through magnetic sensors the position of all the fingers in the arm and hand. So what that looks like in light is like this. The monkey's not wearing the glove in this case, and the light is remaining on during the movement, so we can see what the monkey's doing. You can see the monkeys are very good at this, and they do it very quickly. So while the monkeys are doing this task, we want to observe the internal states of neural activity in the brain. So to do this, the monkeys were implanted with 6 floating microelectrode arrays with 32 channels each. So this is one shot of the brain where you see 6 arrays implanted in the 3 areas that I mentioned. And with these arrays, you're able to record an average of 400 neurons or units at the same time. And at the same time, because of the glove, we can record all the movements of the fingers and the arm in 27 degrees of freedom. And this can be further transformed into a muscle model, which is scaled to the monkey's arm. And from that, we can extract the length of specific muscles, 50 specific muscles. So this is not the same as muscle activity. We haven't recorded the muscle activity in each muscle. But with this, we have access to the muscle length from muscles that would actually, in the hand, that would be very difficult to record. And the signals that you get bear a lot of resemblance to muscle activity in some cases. And you can do that in single trials, so that's what this looks like, slowed down. You can see single trials of the monkey grasping and lifting. So together this gives us a very rich signal of the internal state and also a very rich signal of the behavior that the monkey is doing. So how can we relate this to the network architectures that we mentioned and the tasks for optimization that we already know that certain parts of the brain are doing? So the first thing that we did was we took kind of an off-the-shelf CNN that's already trained for object identification, in this case VGG. We generated images that are simulated images from the monkey's perspective. So this is something like what they would have seen for grasping a large ring. We fed them through the CNN, didn't train it any further, we just fed them through, and then we looked at the individual layers along the CNN and tried to predict neural activity from activity in those layers for each of the 3 areas that we were recording from. So we look at this for a measure that's normalized by the differences between areas, so we can compare between areas. What we see is that as we go along the layers of the CNN, so from pixels, so just the raw image, to the final layers that tell you what object is in the image, we see 2 things. One is that the ability to predict neural activity increases with layer, and it increases most for AIP. So it seems actually that the kinds of information in those later layers might be reasonable inputs for AIP. And actually we know that many areas late in the ventral stream actually send information to AIP. So this is actually quite reasonable. On the other hand, if we take the muscle velocity in this case for the monkey while during the movement period, this was during the cue period where the monkey sees the object and we try to predict neural activity from that, we see the opposite effect. So M1 predicts the muscle velocity very well, F5 less, and AIP less so. So there seems to be this reverse representation showing that there might be some transformation from this more visual representation early on to the representation that's more similar to the kinematics that you actually need. So based on these results, we designed a particular network architecture to optimize this task. So 3 modules paralleling the 3 areas that we're recording from. The input module receives activity from high layers of the CNN during presentation of the different objects. And the output module is supposed to generate the muscle velocity commands that the monkey did on individual trials. There's a hold signal for the entire network that tells it basically when to go. The intermediate module is in between those 2, and we had sparser connectivity between the modules then within the modules. So there are a couple knobs that we controlled here. So the knobs that we kept constant are there are 300 neurons total, so 100 in each module. They're trained to produce this time-varying muscle velocity signal, and then there are a bunch of knobs that we tweaked in a number of different ways, actually training over 1,700 different networks. So I'm going to show you the results of what those tweaks were. But basically, type of input, type of output, whether we have modules at all, do we actually need modules at all? How much connectivity do we need to have between the modules? Do we want to penalize certain things in the network, such as make firing rates low or make input-output connections large or small? And by tweaking these knobs, we can see which constraints are actually more consistent with the behavior and with the neural data that we observe. So for this network that I mentioned here, with these inputs and outputs and sparser connectivity between modules, actually able to train very well, and most of the networks we trained were able to kinematically do this task. This is an example of inputs for one trial. So this is a visual input where this is the representation of the specific object shown on this trial. That's a visual go cue, and this is a hold signal that tells the network when to move. And here you see for 3 muscles, the example average muscle velocity signal that it was supposed to generate, and the one that it did. The different colors represent basically different types of objects, and repeated lines of the same color represent different types or different sizes of the same object. And the networks were able to reproduce those movement dynamics very well, and also keep the output 0 before the movement was cued. So,
a short question.
Yes.
In terms of training, like how much training was required to get to this training?
Not that much. I mean, once I didn't train the network on every single trial that the monkey did, so I actually just gave the network kind of random examples with different lengths of these different periods, so random different lengths. And actually, for each condition, I only gave each network 2 examples. So I was able to limit it to about 84 examples for each network, and that made training 1,760 networks feasible, still difficult, but also feasible. So if we look inside the network, I just want to give really short examples of what the neural activity looks like. So what you see on the top is an average firing rate curves for 3 neurons in AIP F5 and M1. So here in the gray is when the object was presented. Between is the memory period where the monkey's waiting, and this dashed line is when the monkey started moving. And here are 3 example neurons from the simulation that are very similar. And what we generally see, although it's very varied, is that, for example, AIP units that have strong visual responses sometimes sustain over the trial, sometimes not. F5 seems to show selective and fluctuating activity during the memory period and movement period for each object. M1 has the strongest representation during the movement itself. And you see actually all kinds of things in between in each area. So while this is useful to see that there seems to be stuff going on in the model that's similar to the neural activity, it's not going to give us a picture of what the network is doing as a whole. So to address this, we used a specific method for comparing the entire population of neurons in the model to the entire population that we recorded, called Procrustes. And basically what it's doing is it's equivalent if you had 2 objects and you wanted to figure out what's the optimal way to rotate one object so that it aligns on top of the other. Can we find a way where these things look similar without distorting them, without making the water bottle wider or thinner? Then Procrustes allows us to do that. And if we do this across the population, so that's what I'm showing here. If we take all the areas in our model and we try and get them to fit onto the neural data and see how much variance we can explain, and then visualize that in the dimensions of highest variance in the neural data. Here's dimension 1, which basically shows a huge change in the movement. Then we're able to extract similar dimensions in the model. So here are just a few example principal components, so dimensions of highest variance. And what we can see here is that the model is quite well able to show similar dimensions. So even though the model was not trained to reproduce neural activity in any way, only to optimize the task, this type of model was able to explain around 65% of the variance in the neural activity across areas. And we can see that it captures both these complex temporal patterns of activity, both before the movement and during the movement, as well as the specific condition features that differentiate by object. So I actually think this is really powerful that we were able to see this without training the network to reproduce the neural activity at all. One other interesting thing that comes out of this, and is an important question, is do the modules actually look more similar to the areas we expected them to look like? Or does the input module look like AIP and F5 like intermediate, or something else? So the first thing we needed to look at was actually how similar are the areas in the brain to each other. So we used a similar method to compare areas within the brain, and what you see here is that basically this is the similarity of M1 to itself, F5 to itself on the diagonal, and AIP to itself. So areas are most similar to themselves, and then kind of intermediately similar to the other areas. So we see this kind of inter-area hierarchy of similarity, and then we want to see if we use the modules to predict each area in the brain, do they actually predict the areas that we thought they would? And if you do this for that example model, what you see, for example here, the intermediate module in F5, F5 is actually predicted best by the intermediate module, so you see that similar hierarchy. And that's really interesting because we never told the intermediate module that it should have an internal state similar to F5. So this is also a really good test of whether the task we optimized and the architecture we had actually showed us about the function of the individual areas. So a couple other things we can test. I mentioned the sparsity between the areas. So actually if we turn that knob, if we make connectivity between areas more or less, how does that affect the similarity to the neural data? So in the example I showed you, the connectivity between areas was about 10%. So with the same metric, if we look at how similar the modules are to the areas versus the areas to themselves, if we fully connect the modules, we see that the mean of this correlation in inter-area hierarchy is much less than with 10% connectivity. And with 1% connectivity, it's still pretty good, but it's actually best for 10% that we used. And actually, if you look at anatomical studies, tracer studies, this kind of intermediate level of sparsity between areas seems to match pretty well, at least for what's predicted in the primate. So if we look at different knobs, if we change things about the network architecture to see if there's other possible networks that could have fit the data better, So here's the network I've been presenting you. So here's some other examples. If we take away feedback from later modules to earlier modules, then we see that the fit to the neural data is worse. If we change from the visual input to just a labeled code that just labels each condition, the prediction is worse. If we shuffle the output conditions so that the kinematics are not matched with the correct object, it's worse. If we get rid of all the modules and just connect everything, it's worse. Or if we keep the sparsity that we had here overall, but get rid of the specific modules, it's also worse. And we did this for a couple different metrics to show that actually this model fits best, better than the alternative models. So with 10% connectivity and this visual feature input and also feedback connections between modules. So last thing I want to show you is, I mentioned that lesioning different areas produces different results depending on which area you lesion. So here's an example. This is a monkey trying to grasp a groove in a plate. This is where they grasp. This is when their hand is extending to grasp. And if you do a temporary lesion to AIP, what you actually see is the pre-shaping of the hand kind of goes away, and often the hand shape is then mismatched, so they might not be able to get into the groove, for example, in this case. So we were wondering if any aspects of our models could explain things like this. So what we did was targeted lesioning. Basically, we took our models and we silenced different fractions of units in each of our modules, simulating a lesion. And we actually found that it was able to produce interesting results only in the cases where we had modules and where firing rates were penalized to be small, meaning the network was penalized for having neurons that were firing much more than they needed to. And we assessed this in a couple different metrics. So if you silence 1% of the units versus 20% of the units in the different modules, one thing you see is that there's more instability if you lesion the output module, so there's more premature movement activity. The kinematic error scales with the module, so if you lesion EIP, you don't see as much error in the movement as you do if you lesion the output module. The movement amplitude is very much decreased when you lesion the output module, and this is similar to previous studies showing that the overall complexity of the movement and the amplitude of the movement goes down when you lesion M1. And the most interesting for me is that if you lesion the output module, the error just kind of, the movements get kind of weird. But if you lesion AIP or F5, what you actually see is the movements seem to become more generic. So the shaping of the hand becomes less appropriate for the object and kind of just more generic in hand structure. So what that actually looks like, for an example of lifting a cylinder. So this is an example of what it looks like to lift a cylinder. These are the muscle velocity dynamics for the different muscles. If we silence 20% of the units in AIP, we actually see the output actually looks still quite complex. And what it actually is doing something more generic and more similar to lifting a ball or a cube. Similar things when you lesion the intermediate module, but when you lesion the output module, what you see is generally the movement kind of degrades, the amplitude becomes less, and the complex features are lost. So we actually see quite a difference between the different layers depending on the modules, and this actually seems to parallel a lot of previous lesion studies. So to summarize that, the network successfully achieved working memory of the task and produced the required muscle dynamics. Even though the network wasn't trained to reproduce any of the neural activity, the internal dynamics of certain networks matched the recorded data better. Specifically, modular networks with visual feature input and sparse intermodule connectivity best fit the grasping circuit and outperform these other control models. And targeted lesioning of modules reproduce a lot of the behavioral deficits observed in previous studies. So we think that this works an important step in identifying the roles of these different areas during grasp control. And there's a lot more details I couldn't show you. You can check out the preprint. The paper's still in review, but hopefully in the near future will be out, and we also want to put a new version of the preprint up soon. So to give you an idea of what we can do with this framework going forward, something we're working on is a toolbox, a deep learning toolbox for training biomechanical control in more complex tasks. So what this toolbox actually will allow us to do is to train all kinds of neural networks to control realistic biomechanical models. Here's some of the knobs that we have. So the complexity of the model, the properties of the muscle, time delays between neurons, noise, the complexity of the neural architecture, what cost function is being optimized, the task, and the order and complexity of training of the task. And here's an example of this specific neural network architecture, so an RNN with different modules trained to do a delayed reaching task. So it's controlling these specific muscles, which are modeled to be pretty realistic compared to human control. When there's a red target, it has to wait, and when it gets a green, it's supposed to move to the specific target. And this is just trained in a continuous manner, so you can just provide new targets and it will keep moving the arm properly. And we think this kind of environment will be very useful going forward to understand complex behaviors such as reaching and grasping. So tying that back to what I laid out earlier on, this is what I think that kind of looks like in terms of how we do science with this kind of deep learning framework. So in the lab, we tend to use the kin-arm exoskeleton a lot, which is a robot where people can sit planarly, they put their arms and they can move in virtual reality, and we can also provide forces at specific joints that move their arms around in space while they do tasks. So this is what I see that looking like. We have a research question and hypothesis. We can generate experiments that we test in the human, where we can measure behavior and maybe muscle activity as a measure of internal state. We can do similar things in primates, in a similar exoskeleton that we have for the monkey. We can measure the behavior and also maybe some of the neural activity. And then we can also build models that match to that specific task, analyze the behavior, and also analyze the whole internal state and try many different forms of these models, compare all of these to be able to draw conclusions about our hypothesis, revise, and start the whole process again. So I think this is going to be a very useful framework over the next couple of years to understand especially complex motor control questions in neuroscience. But overall, I think we have a long way to go to understand behaviors like this in the human, and that's also why I think grasp control is such an interesting topic of study for us and is going to provide results for a long time. So with that, I'd like to thank contributors, especially my PhD supervisor, where most of this work was done, Krishna and Andrew, my postdoc supervisors, the other people on the projects, the technical support, and of course, total funding. And for your attention. Thank you.
We certainly have time for questions. Would you like to start? Okay, I'll go over here and then we'll pass.
Great talk, Jonathan. So I'm wondering, the arrays that you show where the floating arrays, do you have any control of the layer that you're recording from? So one, let me just tell you what I'm worried about. I'm worried about that we are treating the cortex the same way as these convolutional neural networks, but there is a single cell type, and it is some computation that happens, it's just happening some kind of lateral connections. But the cortex has 6 different layers. So is there any insight that you have into that? Are you trying to assess that somehow?
Yeah, so in this data, we don't have any information about the layers. The arrays go down into the sulcus in many cases. So they're electrodes of many different lengths, so they kind of sample all over the place. But I completely agree with you that there's a limit to studying recurrent networks by doing this kind of layout. Because in the actual brain, where each area has its own layer structure, there's a lot more going on than just a ball of neurons connected to another ball of neurons. And that's something that we're very interested in looking at in the future, is recording layer-wise in some of these areas and adding some of that detail into how we model it, right? And that's the power of these architectures, is we can add more or less of that detail depending on whether or not we think it's useful.
Basically, exactly the same question that I also had. I mean, how important are the layers, basically? Maybe one other thing, how important would be subcortical areas for this?
Yeah.
So let's say basal ganglia, for example, thalamus. Do you need that loop? Yeah.
Probably very important. So one thing that you saw in the network that I presented here, there was a hold signal that told the network when to go. Mm-hmm. So I've kind of externalized something about deciding when to move that is very important. And I didn't want to address that in this specific one because we don't really have a way with just those cortical recordings of addressing which one is best. But I definitely think that including subcortical structures into the model is very important if we have access to neural data about these areas.
Great.
Maybe just a question related to flexible responses, so that the subject responds differently to the same object depending on the task context. Is that something that you think would be easy to introduce in this model?
Yes. So maybe I'll just very quickly go back to the layout of the network. So we didn't do it here. I mean, so one thing about this intermediate module is it's not very distinct except that it sits between the other two. But in reality, the area that it's meant to model well, F5, received like lots of prefrontal connections, for example. And in this task, monkeys tended to grasp the object the same every time. So it's kind of a feed-forward task.
Yeah.
They see the object, they do the same thing. And occasionally, they actually did do different things to the same object. And we actually saw that that seemed to reverberate throughout the whole, every area. So I think in future studies, if you have a task where there's maybe a specific rule, like in this
Right.
context do this, in this context do this, I think just adding a connection which goes, for example, to the intermediate module to kind of control those different contexts would actually be explained already really well by this network. We just didn't have that specific data.
Yes.
There we go. Absolutely.
I had a question about the neural variance explained. So at some point you show all the models with the neural variance explained. It's sort of a conceptual question. At what point am I supposed to believe that how much of the variance has to be explained before it's a good model? And how small can the differences be between models before I believe that that difference is meaningful? Meaning that even your least
Small.
realistic model only did a few % worse than your most
Yeah.
realistic model. Do you have some insights onto that?
Yeah, that is a big problem in the field, is how do we know when networks are doing something similar to another network? And I'm using, a comparison metric that I think is useful, but some of the things I didn't show are really, what are the different predictions of these different networks, right? So I showed you the example of lesioning. It's those networks make very different predictions of lesioning, so that's something we can test. Another thing I did in the networks is to actually look, what's the strategy, what's the computational strategy the networks are using to do the task, which is something we can also use to make predictions. So I think to just show something like percentage neural variance explained on its own is not in itself a very useful metric. And I could build you other models where it only explains 10%. But I think it is an important kind of step along the way of trying to capture what might be going on dynamically there.
Maybe,
Yeah, good question.
maybe one point related to that, because in visual research often what people do, which is also not optimal or ideal, but they ask how much a particular subject, either a monkey or human, can explain in terms of variance in another monkey or human. So is that something that is also common in the field of motor control, or is that something that you wouldn't expect to be that consistent? Because otherwise that could provide some kind of reference.
I would expect to see lots of similarity there, but unfortunately with monkeys, it's not very feasible to have
Yeah.
a lot of different subjects.
Yeah.
I think, it's not for motor control, but the approach taken by the Bashivan paper I mentioned of generating new stimuli on the fly for that individual that make neurons go up or down is an example how you can really take the predictive power of your specific network and test it in that subject. And I think something like that for the motor field, like can we generate a particular perturbation or behavioral stimuli, like go to this place at this time, that can actually do something similar. It's a bit more difficult problem because it's not just a static image, but I think those tools could also be very useful.
This might be a little bit unfair to you, but it's just happens
Okay.
when the human is turning the cube, I think that the guys, they just look at the cube and they look at the configuration of the cube and then they take a little bit longer. So what do you think that they are actually, obviously from my point of view, what is happening there is that they are imagining what is the path that is going to be. But is this path that they're imagining a representation, that they have a visual representation of the different states of the cube, which is presumably somewhere in the prefrontal cortex or somewhere in the parietal or somewhere, some other that is visual? Or do you think that is a representation of the sequence of movements that they have to do to actually to complete the cube? I would bet for the former, but I would like to hear what you think about it. So basically they have this set of configuration and they do whatever they have to do with the hand to do. which since they have visual feedback. But it's not really a sequence of movements what they're rehearsing. It's the different states of the cue, and then the movements to get there. It's unfair because that's what the monkeys are doing. But I'm curious about what do you think about this?
I'm not sure what the exact question, but one thing they do is there's also blindfolded competition. Also for one hand and for two hands for Rubik's Cube. It takes a bit longer, I think about 18 seconds for world record level. So it does take them a bit longer without that visual feedback. But basically they are executing a planned sequence, right? So what you saw in the beginning of the video is he had 15 seconds to look at it, right? In that time he was planning every move, and then in the 10 seconds that he did it, he was just executing that sequence of moves, right? Which is actually similar to what the robot I showed you was doing, because the robot knows what sequence of moves it wants to make, and then it's just trying to do those sequences. Whether it's more related to the visual representation of the cube or the sequence of physical movements, I'm not sure.
But I think it's worth pointing out the hard part is actually moving the darn cube.
Yeah.
Figuring out the sequences is easy. Any simple computer algorithm can figure out how to go from any arbitrary place on that Rubik's Cube to the final. So that sequence is super easy.
No, but not a human. After 5 of these states, you're done.
But I'm
Working
saying,
memory is
but I'm
outdated.
saying a computer can easily write out the state of transformations required to go from A to B. But even the most sophisticated robot can't actually execute it with that hand.
I think what you're saying is that one thing that's difficult about that is you have to keep track of where you are in that sequence and of the order of the sequence. So even if you know the maximum 20 move sequence, actually keeping track of what you're doing and doing the right thing is.
I think the guy has to say, okay, the greens has to be on top, the ones has to be there, and not exactly, oh, I have to rotate that, I have to do that. The blindfolded, because if there is a prolongation of time, but if they can do it, it's definitely not that. If they can do it, they're definitely doing something with the sequence.
Yeah,
but they're
yeah.
helping with the visual, maybe they're just loading chunks. I get it to that place, and then it's very hard, it's fascinating to me that they can do it with blindfolded. That's absolutely
The
crazy.
confound here is that when it takes us a long time to solve it, like me, it takes me a long time to solve it. Most of that is because I don't know what move to make.
Yeah.
I can move my hand perfectly fine, right? I can do the movements, but I don't know what movement to make. So that performance you see there, of course, obviously, a lot of it is that 15 seconds where they have to figure out the moves, but it's also on its own incredibly impressive that they can take that sequence and do the whole thing in 10 seconds. While for us, probably we look at this, and if you haven't done it before, the hardest thing for you is you don't know what to do. If someone tells you to turn it, you can turn it just fine.
So how many more moves to actually complete that cube?
This one,
Yeah.
if I knew exactly what to do, probably 5 or 6,
Wow, okay.
but I don't. The maximum is 20, so from any position, the maximum number of moves is 20. So.
Wow. If there are no questions off-site, if there are no more questions today, please join me in thanking my colleagues for the
You can memorize the space of the after working memory capacity is just it's gone. So I just say, okay, I bring this up one side.